Okay, let's get on with lesson 1 of OCR Human Biology AAQ. This is a level 3 course, so I'll try my best to go over all the level 3 content that you will need to know for this course. First thing we're going to consider is, what is a cell? Well, a cell is the smallest unit of life. So think of a little cell like this. That is the smallest unit of life. This is an important definition that you'll need to know when you're asked in an exam. What is a cell? It's the smallest unit of life. Now, there's different types of cells. There's eukaryotic cells and prokaryotic cells. Now, let's look at the difference between those. A eukaryotic cell, as you can see, is a lot more complex than a prokaryotic cells, but there's a main difference that you need to know in your exams. For one, a prokaryotic cell does not have membrane-bound organelles, whilst a eukaryotic cell does have membrane-bound organelles. So let's break that down. This, no membrane-bound organelles. Well, this does. Has membrane-bound organelles. That is one major difference. Another major difference is that eukaryotic cells are much bigger than prokaryotic cells. So we also want to talk about how eukaryotic cells are bigger. And prokaryotes are smaller. These are two major differences that you can probably list in your exams, and they're very easy to remember. Think about the fact that a lot of prokaryotic cells are bacteria, and bacteria is small. Eukaryotic cells are things like your body cells, like your skin cells, even your blood cells. Well, red blood cells have a special difference as well. But a major difference as well is that a eukaryotic cell has a defined nuclear region, as a defined nuclear envelope. What I'm circling right now is the nuclear envelope. Prokaryotic cell doesn't have that. It's messy. All the genetic information is displayed about. That's called the nucleoid region. And there's also an extra ring of DNA called the plasmid. That's important for, cell replication in bacteria, but that's for a different lesson. So those are major differences between a eukaryotic and prokaryotic cell. Now let's talk about cell structure. As you can see, this is pretty complex. So I'm going to use a GCSE model to kind of make it like a bit simpler. Now you can see it's a lot more different than the level 3 diagram, but we're going to go over the level 3 organelles that you will need to know. So as we know, an organelle is just kind of like, the little organs inside the cell, the little tiny little organs within the cell. The first thing that we're going to talk about is the cell surface membrane. You can see it kind of drawn as a line, but that is inaccurate, because the cell surface membrane is actually a phospholipid bilayer. So that's important to write down. A phospholipid bilayer. It's a phospholipid bilayer. I even exclaimed it because it's very important to know that. This phospholipid bilayer is embedded with different proteins. So let's also write it down. It's embedded with proteins. I'll go over the biology of the phospholipid bilayer another day, but that's basically a very important thing you need to know. Now, the function of the cell membrane is to let certain things in and out. That's why we call the cell membrane semi-permeable. So let's write down that the cell membrane is a semi-permeable kind of semi-permeable. It's a semi-permeable kind of membrane. Kind of membrane. It is a membrane. It's semi-permeable, and it allows certain things to come in and out. So, like helpful nutrients for the cell can go in, while, harmful things that want to enter stay out. Pretty self-explanatory. Now let's talk about the cytoplasm. That's another thing you might remember from GCSE. The cytoplasm is a jelly-like substance. So let's write that down. A jelly-like substance. Jelly-like substance. It's like a little bit gooey, like that. So this is actually the site of metabolic reactions. I don't know what you were told at GCSE because I have not done my GCSEs in ages, but it's kind of like a jelly-like substance that is the site of metabolic reaction within the cell. So let's write that down. The site of metabolic reactions. The cytoplasm is kind of also filled with, like, special enzymes that help it with this function as enzymes. So if you can remember from maybe GCSE, if you're just starting human biology, so enzymes are kind of like catalysts, So they help with catalyzing the process, like the metabolic processes within the cell. It contains certain organelles, like ribosomes, as we can see, the little ribosomes, and, dissolved substances, basically. Now, branching on that, let's go to the ribosomes. You may remember ribosomes as being the site of protein synthesis, which still holds up at level 3. So let's write that down. The ribosomes are the site of protein synthesis. Little smiley face. They're the site of protein synthesis. Now, a major distinction that you need to know at level 3 is that in eukaryotic cells, they're not just the ribosomes are bigger. So we say that they are 80. Oh wait, let me get my pen. ATS ribosomes. So these are ATS ribosomes, which basically means they're bigger. And the nucleus like contains all like the genetic material within the cell. So let's write that down. Oh, I'm gonna use a rainbow pen. Where all the genetic information is stored. Let me get serious. Let me not use a rainbow pen. But anyway, so the nucleus is basically where all the genetic information is stored. Now, genetic information in a cell, or in anything actually, genetic information we know that as DNA, deoxyribonucleic acid. Now this is just a level 3 diagram, so I want to actually go deeper into the nucleus. So this is kind of a detailed level 3 diagram of the nucleus, and it's like the nuclear envelope. So as you can see, nuclear envelope has little pores. You can say that the nuclear envelope is porous. These pores allow things like mRNA to, leave and come back, come back to nucleus. So basically, within the nucleus, we also have chromatin, which is, little squiggly stuff. So the chromatin actually forms, later on, chromosomes when it condenses. That's important to know. Like, so the chromatin condenses into chromosomes. Now, what we also need to know, the nucleus is also kind of charged with, like, gene expression. So you'll have certain genes that need to express and certain things that stay not expressed. Certain things that just don't express. I think this is basically what you need to know. Now, not all eukaryotic cells actually have a present nucleus. For example, let's draw a little red blood cell. For example, red blood cell. You know those little things? Those red blood cells, they carry your oxygen. They don't have a nucleus. Now, this is a specific adaptation because the red blood cell wants more space, more surface area to grab onto the oxygen to transport it around your body. So, mature erythrocytes, aka red blood cells, don't have a nucleus. So that's an important thing that you need to know for your exams. Maybe the example will ask, hey, what adaptations do the red blood cell have to carry oxygen? They have no nucleus. They are foregone all the organelles for some reason, but more power to them. Anyway, useless little side tangent. Let us go on to the mitochondria. So an important thing that we need to know about the mitochondria is that they are the site of aerobic respiration. The site of aerobic respiration. So, aerobic respiration, I like to remember aerobic respiration as go cow. So glucose plus oxygen equals CO2 plus water. Now, another thing that the mitochondria does, apart from aerobic respiration, is produce ATP. ATP is very important. You can kind of think of it as the energy the cell needs to do its functions. Now that we've gone over this kind of example of, a level 2 diagram of a cell with some level 3 additions to make it more digestible for you, we can go on to a bigger cell like this. We've already went over the nuclear envelope. We've went over the fact that the cell membrane is actually a bilayer, a phospholipid bilayer. We can go on to more, a bit more complicated definitions. Now, this is the rough endoplasmic reticulum. As you saw, it was surrounding the nucleus here, but this is the rough endoplasmic reticulum, and we also have a smooth endoplasmic reticulum. Now, let us go to my little diagram here. So this is a rough endoplasmic reticulum. As you can see, it's studded with these little balls. These little balls are actually ribosomes. They are studded with ribosomes. Fashion trend, I suppose. And it synthesizes and processes different proteins. You can kind of think of it like that. It basically synthesizes and produces proteins. So let's write that down. Synthesizes and processes proteins. Sorry for my abysmal handwriting. Processes proteins. It kind of processes proteins, and these proteins get secreted into your body. So these little things here, it processes proteins. So these ribosomes here, they process proteins that are going to be secreted. So processes to be secreted. Now, what I mean by secreted, they kind of exit the cell, basically. So you kind of can think of it as the editor of your proteins. Now, when we think of the smooth endoplasmic reticulum, as you can see, it's not studded. It's not studded. It's smooth. It's completely smooth. And its function is basically to synthesize lipids and steroids, and, it detoxifies. It also stores calcium ions. But the main thing that you want to know is that it basically synthesizes lipids. Synthesizes lipids. That's basically a basic rundown of what it does. I do implore you to do your research and not just watch my videos. So that's the main differences. So as you can see, rough. When we're talking about rough, I like to remember the RER. I just, instead of rough, I say ribosome. So this is the RER. You can remember it's studded with ribosomes. And this is smooth. So this will be the SER, the smooth endoplasmic reticulum, and it's smooth. Remember that. That's the main difference. Smooth and rough. Studded with ribosomes, no ribosomes. Now we're back here. The Golgi apparatus. The Golgi apparatus is kind of like they're little flattened sacs. It's kind of like a membrane. It's like a kind of network of flattened sacs, like membrane sacs. There's the cis face and there's the trans face. I don't know why they called it that, but and it basically kind of modifies and packages sorts proteins and lipids, and produces like vesicles and lysosomes, which we'll talk about later. So you can say it kind of packages up proteins. Like it just packages up proteins and lipids. So packages proteins and lipids. Yay! So that's basically that's the basic thing of what it does. Do your research. I'm just kind of summarizing it for you. So here we have lysosomes. Lysosomes are like membrane-bound sacs containing like enzymes. Like acid hydrolases, just enzymes. That has enzymes inside of it, right? And it digests worn-out organelles, pathogens, and cellular debris, and they are formed by the Golgi apparatus. So basically, they eat dead stuff. So you can kind of say that the lysosomes, they digest and hydrolyze dead organelles, debris, or pathogens. So hydrolyzes, I'm probably not spelling that right, hydrolyzes and digests worn out organelles, pathogens, and debris. Yeah, that's basically what you need to know. We already went over the mitochondria, kind of already went over... Oh, I realized I've not talked about the nucleolus. Okay, so the nucleolus is this dense sphere that you see inside of the nucleus. Like when you look at a picture under an electron microscope of, the nucleus, you'll see this little dot. This is the nucleolus, and it produces ribosomes. That's the basic answer for you. It produces ribosomes. Little dots there? Yeah, it produces those. And you know what ribosomes do? The site of protein synthesis. Now, the last thing I want to go over is kind of the structure of mitochondria. So, mitochondria, some important things that you need to know, they have a double membrane. So, a double membrane. You know that they have a double membrane. They have like, and the inner membrane is folded into these kind of sister, like cristae. I think that's how you say it, cristae. So the inner membrane here is folded into these cristae. I think that's how you say it. And they kind of help with creating like a bigger surface area for, you know, respiration, chemical reactions, for creating ATP, which is essential for cell processes. Now it kind of has like a fluid filled matrix. Now the matrix, it contains like enzymes and things that kind of help with chemical reaction, and it has its own ribosomes. Now these ribosomes are smaller than the ribosomes that you see in, you know, the whole cell. So these will be called 70S ribosomes. Oh, you don't see that. So the 70S kind of means they're small. They're smaller. That's important. That's important for you to know. And they have their own DNA. Like their own little DNA, kind of like a prokaryotic cell. And that's because a prokaryotic cell, wait, where did I put it? Where did I put it? So a prokaryotic cell and a mitochondria are actually very similar because they're kind of one and the same. But that's for a future video. Now, I hope that you learned something from that. And thank you for tuning in to my very chaotic teaching of cell structure. I hope that you learned something from it.